Hello everyone, Namaskar to one and all. This is Chapter 10 of Unit 3. This chapter is Cell Cycle and Cell Division. This is Part 1. It includes cell cycle, detail of cell cycle, that is detail of interphase, G1, S and G2, and then we have to discuss in the last, that is G0 phase. This is Part 1st, Chapter 10. Children, you know that all organisms, either this is the largest or the smallest one, made up of single cell or many cells, that is, they are being derived by single cell. Growth and reproduction are the characteristics of cell and is found in all living organisms. All cells, they reproduce by dividing into two. Each parental cell gives rise to two daughter cells each time when they used to divide. You know it. The newly formed daughter cells, they grow, divide, giving rise to, that is, new population, and in that way growth and division of single parental cell make the progeny. Such cycle of growth and division allows single cell to form structure and consisting of millions of cells. You know, in our case also, that is, we start our life from the single cell, that is zygote, having 2n number of chromosomes. It divides into two, having same genetic constitution, and this division, that is, it goes on, and ultimately a multicellular body having trillions of cells, that is being formed. So this is the same pattern we have to see in all organisms. Now we will discuss what we mean by cell cycle. You see, cell division is very important, found in all organisms, as I told you. What happens during cell division? There is DNA replication and cell growth take place. DNA replication and cell growth. All these processes, cell division, DNA replication, cell growth, all these processes, they take place in a coordinated way to ensure correct division and formation of progeny that containing intact genome. Important one here is all these sequence or events in a sequence by which cell duplicate its genome, synthesize other constituents of cells, and eventually the cell divide from one cell, that is two cells are being formed, that is known as cell cycle. So during cell cycle what happens? There will be duplication of cell. From one will get the two cells, and in this there will be a sequence of events, that is DNA will be duplicated, and all other constituents of the cell, they will also be duplicated, and ultimately the parent cell will divide into two. That is, cell growth in terms of cytoplasmic increase, that is a continuous process growth. Cell used to grow. DNA synthesis, that is, it doesn't happen in all these stages. A particular sequence is there which we have to see in time to come. So we have to see now, that is replicated chromosomes. You see, that is duplicated DNA will be distributed to the daughter cells, and all this will be under the control of genes. Genetic control is there. I told you in cell theory that he was the first one, Rudolf Virchow, who suggests that a new cells are formed from the pre-existing cell. The statement given, you know, a very popular statement, omnis cellula e cellula. Now this is important aspect. One may ask, why cell has to divide? What's the reason? You see, during growth what happens, that this is a young cell, this is the nucleus, that is, it will grow. The size of the cell, volume of the cell will increase, but in proportion the nucleus will not increase. Ultimately, let the cell has grown to this size, and this is that is nucleus. You see here, if you say that is volume of cell and that is surface area, ratio of surface area and volume. When the cell will grow, children, you know, that is surface area will decrease. That is, in a young cell, surface area will be more, and in a cell which is fully grown, the surface area will be less. That's why what happens during this growth, that is ratio of surface area and volume that decreases, because the surface area that will be less and the volume will be more, the ratio will decrease. So see here, that is to maintain this nucleocytoplasmic ratio. One more thing happens here, when that is the cytoplasm content will be more, that is the control of the nucleus will decrease. Therefore, to maintain this, that is nucleocytoplasmic ratio, cell has to divide. The nucleocytoplasmic ratio is also called as karyoplasmic ratio, current plasma ratio, that is to be maintained, and to maintain this, cell has to divide. Right? Come to the next now, we have to see phases of cell cycle. That is, a human cell in culture, the cell cycle, it is approximate of 24 hours. However, duration of the cell cycle, it will vary from organism to organism. The yeast, for example, it has life cycle, that it has cell cycle only of 90 minutes in case of yeast. M phase represents the phase when actual cell division takes place, that is, the cell will divide into two. That is known as M phase. It is significant to note that out of 24 hours in case of human cell division that lasts only for about 1 hour. If this is the total, that is cell cycle in case of human, the total cell cycle is of 24 hours. Out of this 24 hours, this phase, when the cell actually doesn't divide, preparation of the division will take place, it is of 23 hours, while that is the M division, M phase, in which cell usually divide into two daughter cells, that is only of 1 hour. Therefore, the interphase, this is interphase, it is about 95% of the total cell cycle, while M phase is only about 5%. Division of the nucleus, that is denoted as karyokinesis, it will take first, and it is being followed by division of the cytoplasm, that is cytokinesis. You see, interphase, traditionally we used to call resting phase, but this is misnomer, misleading one. Cell is never at resting phase. In this phase, cell used to prepare itself for the next cell division. Here will be growth of the cell and DNA replication. Therefore, the cell at this stage is not at rest. This is metabolically, that is highly active. You see now, the cell cycle we can divide into two phases, interphase, which is about 95% of the total, and M phase, which is 5%, 1. We have to see now, that is interphase, detail of interphase. As I told you earlier also, this is the M phase. The newly daughter cells have been formed. Now these daughter cells, they will enter into interphase, and ultimately during this interphase, that will prepare itself for the next cell division. So see, this interphase is also known as preparatory phase, and it is the stage between two successive cell division. That is, this cell division is over and now the next cell division is to be started. This is in between the two, that is successive cell division. What is there in this? No division of chromosome or cytoplasm, and what is important here, nucleus and cytoplasm, that will be metabolically very active. It makes about 95% of the total, and this interphase is the longest one, and we can divide this into three phases. One is, that is G1 phase, followed by S phase, and it is followed by, that is G2 phase. We have to see detail of this now. See, first phase of the interphase is called G1 phase, gap 1 phase this is. Post-mitotic gap, why post-mitotic? You see again that was the parental cell. It has divided into two daughter cells. Now that is division, mitosis division is over. That's why this is the cell just after mitosis division, and it has to enter into G1 phase. So we can call that G1 is post-mitotic phase. Right? What happens here? See some important characteristics. There is cell grow to the maximum size because normal metabolic activities will be there. These are very fast because after this they have to enter into S phase, and that's why here this is preparation for S phase. You know, children, in S phase the important one will be there is DNA replication. See, DNA replication will not take place at once. The preparation is required. For replication of DNA, you know, nucleotides are required. Nucleotides, raw material for different type of enzymes are required, and all this will be prepared in G1 phase. Nucleotides, the amino acids, that is which will make the protein, and energy giving ATP molecules will be synthesized here in G1 phase. Important one, cell organelles also increases in number except mitochondria, chloroplast and centriole. All other, for example, that this is Golgi body is there, that this is ER is there, they will be duplicated here. The cell will grow to the maximum size, most variable length, because the time of cell division differ from cell to cell. It is because of that is time of G1 varies. G1 is terminated with various stimuli, but once the cell has entered into G1 phase, after that the cell has to enter into S and the cell has to divide. Can we proceed to the next now? Yes, you see a good diagram is there. That is, this is, what is this? This is amophase. From this to this one is amophase, and from this to this one is, that is interphase stage. This is G1, followed by that is S, and followed by that is G2, and after G2 again that will enter into, that is amophase. You know, you are seeing in the diagram amophase. G2 is being followed by prophase, then it is being followed by metaphase, then anaphase, and then this is telophase. You know, that is these stages the nucleus will divide into two. Let the parent cell was here. Now at this stage the parent cell will have two nuclei. So this division is called karyokinesis, and after this, that is cytoplasm will also divide, so that this cell that will make two daughter cells like this, each is having its own nucleus. Right? Now important one here is what we mean by G0 phase, also called as quiescent phase. Some cells in adult animals, they do not appear, they do not divide, they do not appear to exhibit sort of division. For example, heart cell is there. What is there? That is, they have lost division capacity. Why? Because of injury or that is cell death. Sometimes they need to divide. So these cells, they do not divide further. Usually they do not divide, and they do not enter, they exit G1 phase, and they will enter, instead of G1, they will enter into an inactive stage or quiescent phase called G0 phase. So G0 phase, the cell doesn't divide. Cell in this stage remain metabolically active, but it will not divide till it is being asked to divide. Depending upon the requirement, this G0 that will remain either silent, or it may, that is, it may differentiate, means doesn't divide, or if it is being asked, then it will enter into division. So see here, this is amophase, that is, this is highly specialization is there. For example, nerve cell, it has entered into the G0 phase, and if the need will be there, then it will divide, otherwise it will not divide. Right? Come to the S phase now, which is very, very important one. S phase. What we have in S phase, children? Also known as synthetic phase. synthetic phase. Synthesis of what? Here is synthesis of, that is DNA. DNA replication will take place in this. That's why it is known as synthetic phase. Amount of DNA will be doubled. However, there is no increase in number of chromosome or ploidy will remain same. This statement is very, very important one. You have to understand. I'll tell you the detail of this. See children here, let this is the M phase is over. Let that was the parental cell. See it carefully. This is the parental cell. Let this parental cell is having, this is the chromosome. Let it is having, that is one chromosome is this and the one chromosome is this. Let this is having number of chromosome here is 2. And let each this chromatid, these are the chromatid, each chromatid is having 1C amount of DNA. So here in this parent cell, amount of DNA, total amount of DNA will be 4C. Is it clear? This is parent cell. How many chromosome? 2. DNA 4C. Now it has entered into M phase, mitotic division, and it divided into 2 daughter cells. You know, in mitosis number of the chromosome remains same. So here the number of chromosome is 2. Here also in daughter cells number of the chromosome will be equal to 2. One will be here, this is 1, this is 2. One will be here in this 1 and 2. The number of the chromosome in each is equal to 2. Can you see the difference here? Here number of chromosome is 2, but the chromosome is made up of 2 chromatids. Here number of chromosome is 2, but the chromosome is represented only by 1 chromatid. This is the basic difference. Numerically number of the chromosome in daughter cell is equal to the parent cell, but it is represented only by 1 chromatid. So these are, you see, you have to concentrate on chromatids. Here number of the chromatid is 2 in 1 chromosome. Here number of the chromatid is 1. What will the amount of DNA here? Amount of DNA in one daughter cell, that will be 1 and 1, that will be equal to 2. See, is it very much clear? Now you see that it has to enter into G1 phase. In G1 phase, this is the cell here. Amount of that DNA, this is the chromosome. How much chromosome we have here? 2. How much DNA we have in G1? Again that will be 2C. But you see what will be there in S phase? S phase. S phase also, number of chromosome will be equal to 2. I am going to make it here. Let this is 1 chromosome and let this is the other chromosome. So how many chromosome after S or in S? That is 2. And amount of DNA? Can you tell me? Amount of DNA will be, that will be equal to 4C. What statement is? In G1, number of the chromosome will be equal to the parent cell. But in S and after S, that is in G2, in G2 also that will enter into G2, number of the chromosome will be 2 and amount of DNA will be, that is 4C. Amount of DNA will be doubled here. You see now, very interesting one. Initial amount of the DNA, if is denoted by 2C here, then it replicate to 4C. Is it clear? And the cell has 2 and a number of chromosome at G1. Even in S, number of the chromosome that will not change, that will remain same number of chromosome. Right? Histone protein will be synthesized here in S phase. S phase is called invisible phase because replicated chromosome are not visible at this stage. This will help you to solve so many numerical problems. Many more times with numerical problems also we will discuss it. Clear? See this beautiful diagram we have here. Let children, that is, this is G1. G1 is followed by S. S is followed by G2. G2 is followed by M. And then again that will enter into the G1. See, let this is number of chromosome. Can you tell me? number of the chromosome. Here, for example, we have taken 2 number of chromosome in G1. Sorry, G1 is this one. Number of chromosome in G1 is 2. In S also number of the chromosome will remain 2. In Z2 also number of chromosome will remain 2. And in M also number of the chromosome will remain 2. What's about DNA content? In G1 you see here, this is G1, amount of DNA is 2C, G1. In S, amount of the DNA has increased to 4C. In G2 also, amount of DNA is 4C. And in M also, amount of DNA that will remain, that is, in the beginning that will be 4C, but as and when this M will be over, amount of DNA will be equal to 2C here. And in G1, again that will be equal to, that is, this is the, that is related to number of chromosome, and that is amount of DNA. Very, very important. That is part of the chapter. This one is, you have to concentrate here on this. Now come to the last part of this, Z2 phase, pre-mitotic phase. Why this is called pre-mitotic one? Now the things are clear. This is interphase, this is G1, this one is, that is S, and this one is G2. After G2, the cell is fully prepared to divide. So G2 we may call that this is a stage before mitosis division, pre-mitotic one. This is interval between DNA replication take place here, and this is the mitosis. It lies in between DNA replication and M phase. Is it clear? Actual preparation of M phase will occur here in this, because in phase, M phase, there will be, that is, this is dramatic phase, much more changes will be there, and these changes that they will be prepared in Z2 phase. That is synthesis of RNA, and protein will continue here in this, because here in this, you know, M phase, the chromosome, they will go to the opposite pole with the help of these spindle fibers. Therefore, protein which will make these spindle fibers, alpha and beta tubulin, will also be synthesized to make spindle fibers. An important one here is cell organelles such as mitochondria and chloroplast will also be duplicated here in Z2 phase. That is mitochondria, and that is plastids, or we can call that is chloroplast, that will be duplicated here. And synthesis of protein kinase occurs for regulation of cell division. We have to see in time to come what we mean by this. Right? We see here, we can summarize here again. That is, you can go through some important points again. We have to see, I told you that cell division is important characteristics of all livings, that is growth and reproduction. In case of, that is, unicellular organism, we cannot differentiate growth and reproduction. These are inclusive terms, you know. You must have studied in that is earlier chapter. In case of, that is, prokaryotes, or in case of, that is, unicellular organism, the terms are inclusive, while in case of multicellular organism, that is growth and reproduction, in case of multicellular organism, these are exclusive event. These are exclusive events, right? And that is, population of the organism will increase because of, that is, division in cell. Okay? Statement, that is, they divide, you see here, okay, that is sequence of events in which cell will duplicate and will synthesize other constituents of the cell. This is known as, that is, cell cycle. And who told that cell divides? Very existing cell will divide. That is omnis cellula e cellula. The statement was given by Rudolf Virchow. Why the cell divide? Because to maintain this nucleocytoplasmic ratio or current cytoplasmic ratio, that cell has to divide. Phases of cycle, that in 95% that will be of interphase, about 5% that is amophase. Interphase, I told you, this is not the resting phase, this is metabolically active and is divided into G1 phase followed by S followed by that is Z2 phase. Interphase I told you here now. G1 phase characteristics: cell used to grow, cell organelles except mitochondria and chloroplast centriole, they will be duplicated and there will be increase in that is size of the cell. Then this diagram I told you, that is G1. The question used to come in examination, which is the proper sequence. Proper sequence will be like this, that this is M, then this one is G1, then this is S, then this is G2, and again that will be M. If we want to ask one more thing here, let this is, that is prophase, anaphase, then this will be after prophase that will be, you can see here prophase, after prophase that will be metaphase, anaphase, telophase, then this is karyokinesis followed by cytokinesis, all together that will be M phase. M phase will be followed by this. We have to see it now. G0 phase I told you, S phase, and that is Z2 phase characteristics of this. This is about part 1 of chapter 10. Thank you.